Good morning, my dear friends. Welcome back to my YouTube channel Medical Classes by Dr. Srinidhi Kumar Acharya. Please don't forget to subscribe to my channel and also please give your valuable comments. You can also visit the playlist. In the playlist, I have provided almost all the topics in a very systematic order. Definitely you can make use of it. I hope this will be very beneficial to you. My dear friends, we have been discussing about the bleeding and clotting disorders in children. In that, we have looked into the introduction and basic parts and clinical interpretation of the coagulation profile, clotting factors, platelets, and disorders related to it. Now we have also familiar with the fact that there are two important pathways, intrinsic pathway and extrinsic pathway, how we need to differentiate between these two. And today onwards we will take the disorders of the intrinsic pathway. Intrinsic pathway disorders are very important. It includes a big group of disorders by name hemophilic disorders or hemophilia. So today onwards we shall discuss about hemophilia, which comes under the intrinsic pathway. Now, what is hemophilia? So hemophilia is a disorder of secondary hemostasis. This is a disorder of secondary hemostasis. That means primary hemostasis or temporary plug is different, which is basically done by the platelets and von Willebrand's factor. Whereas the permanent formation of the plug, a stronger plug, is the formation of the stronger plug or formation of secondary hemostasis. That is the function of the clotting factors. So when I say secondary hemostasis disorder, that means this is related to the clotting factor. In the clotting factor also, it is related to the intrinsic pathway. Now ancient Greek words, hema, that is called as blood, and word meaning of the philia is lust or affection. So hemophilia word meaning in Greek goes like that, lust for blood or lust for bleeding. So this is also called as bleeding disorder. Okay, so basically hemophilia is also called as Royal's disease. Why this name came is in the 18th century, Queen Victoria of England found with the genetic mutation in her gene, which is meant for coagulation. And later he started with the bleeding and also later diagnosed as hemophilia. So as it happened or occurred in the royal family for the beginning, the first time when it is identified, so therefore it is also called as Royal's disease. Now this is also called as bleeder's disease. The common hereditary coagulation disorders due to the deficiency of reduced synthesis and action of the clotting factor 8 and as well as 9. Clotting factor 8 and 9 both comes under the purview of intrinsic pathway. Okay, basically in hemophilia there are many types, but most common being factor 8 deficiency and factor 9 deficiency. Factor 8 is also called as hemophilic factor. Okay, so now basically hemophilia is an X-linked recessive disorder. So when I say X-linked recessive disorder, recessive means, as the word suggests, you need both the genes get mutant to produce a disease. If the only one gene is mutant, then it becomes a carrier. When the both are get affected, then it becomes a disease manifestation. X means this is in the X chromosome, that is sex chromosomes. So it is a X-linked recessive disorder. The normal principle says that all the X-linked recessive disorders are very common in the male compared to female, because female has got two X. If at all one X get mutant, the another X will be there, so that will do the function. So the female continue as a carrier and not as a disease manifestation. Whereas in the male we have got only X and Y. If that one particular X is get affected or mutant, then it is something very similar to the dominant condition. And as there is no other X, another one is Y, isn't it? So there is no other X to make the balance. Okay, so and one X which is existing, that is defected or mutant, So definitely there will be the presentation of the disease. So in general, usually we say X-linked recessive disorders are very common in the males, and same is also true with the hemophilia, which is very common in male, and in the female it continues as a carrier. So main sufferers will be male and affected female will be always carrier. But however, there is some exceptions are there. Females usually not show any symptoms of hemophilia. However, the symptomatic female carriers may exist. Symptomatic carriers that means actually she is a carrier but having some symptoms of a hemophilia, that is possible. And sometimes even the female maybe also get the proper hemophilic disease, and that is because of X activation. Mary-Leon hypothesis explains that, so that we'll discuss later. Now, X-linked inheritance in hemophilia, if you continue to discuss, there is a mother with 2 X chromosomes and one is mutated and another is normal. This is what we have already described. Similarly, there is a father with both normal X and Y. Now the possibilities of the genotype is, I think, just like this. Okay, there is an XY male is normal, XX female is a carrier. Then there will be a possibility like this: XX, XY, XH, X and XHY. So if you see in the males, okay, so suppose if this couple will have 4 children, we assume that 2 will be normal, 1 will be male, another one will be female, and both will be normal and they are not affected and not they are carriers also. But one of the female will be a carrier and one of the male will be deceased, because the male is only having X, only one X, that is also defective X, that is mutant for hemophilia, and Y is there, so there is no another X, so he will be deceased. So this is what is called as X-linked recessive pattern. This is typically what is called as X-linked recessive pattern. The assumption is there is a couple and they will have 4 children, and out of that 4 children 2 will be male, 2 will be female. Okay, this is our first assumption. If that was the case, and this is a possibility. Now we cannot say, isn't it, if all the 4 are females only, if these couples will have all the female kids, and this will not work. But for the understanding purpose, easy understanding purpose, we assume like that there will be 4 kids, out of them 2 will be male, 2 will be female. Then this will be the possibility. So now female with the mutated X will be carrier as she has got another X, and she becomes carrier. In recessive disease, both genes must be mutated to disease for the occurrence of the disease, that we have discussed already. Now let us discuss the types of hemophilia. There are 2 main types. One is hemophilia A and another one is hemophilia B. Other types are hemophilia C and also the parahemophilia. But there is one more type which is called as vascular hemophilia and which is also called as von Willebrand's disease. That we will see later. Now hemophilia A, this is because of the deficiency of the clotting factor number 8. So clotting factor 8 is also called as anti-hemophilic factor, and it is discovered first, hence it is called as also classical hemophilia. Among the hemophilia group of the disorders, it is the factor number 8 deficiency or anti-hemophilic factor deficiency which is discovered first. Therefore, this is also called as true hemophilia, or this is also called as classical hemophilia. So the prevalence will be 1 in every 10,000 babies. So therefore, this is the most common type. 1 in 10,000 babies means it's for good incidence. Now hemophilia B, talking about hemophilia B, this is due to the deficiency of the clotting factor 9, which is also called as Christmas factor. Yes. So hemophilia B, this is because of the clotting factor 9 deficiency, and this factor is also called as Christmas factor. Okay, and therefore this is also called as Christmas disease. The first one is called as classical hemophilia, and this is also called as Christmas disease, or this is also called as hemophilia B. The hemophilia A is called as classical hemophilia, true hemophilia, or hemophilia A, whereas hemophilia B is also called as hemophilia B or Christmas disease. Looking into the incidence of hemophilia B, and this is even more rare, 1 in 50,000 or 1 in 1 lakh. So compared to the hemophilia A, hemophilia B is quite rare disease. Come to the next one, that is hemophilia C. Hemophilia C is because of deficiency of the clotting factor 11. Okay, so clotting factor 11 is also called as plasma thromboplastic antecedent. If there is a deficiency of the 11 factor, then it leads to hemophilia C, whereas parahemophilia is because of the deficiency of the clotting factor 5. So the intrinsic and extrinsic pathway, we know that everything is 5, 11, 8, 9. So these are all comes under the intrinsic pathway. Therefore, hemophilic group of the disorders are always because of the problem in the intrinsic pathway. And now coming to the pathogenesis. Earlier, the hemophilia occurs due to the deficiency of the clotting factor 8 or 9 was understood. Previously, we understood that hemophilia is only because of factor number 8 or 9 deficiency. And this deficiency may be due to some lesion in the genes responsible for making these clotting factors. Okay, there are some genes in your body, each cell, which is responsible for making this clotting factor, all the clotting factors. For individual clotting factor, there will be individual gene is there, and in that individual gene is get affected or mutated to some extent, then it leads to definitely the formation of these clotting factors are affected. Rather, genes will going to produce the proteins, and all these clotting factors are made up of the proteins. Now, so such genes are always present in the X chromosome. So they are not present in the allosomes. They are only present in the X chromosome or sex chromosomes. So type of genetic, therefore it is called as the X-linked disorder. So types of the genetic mutations includes deletion, nonsense mutation or inversion. So this is the possibility of genetic mutation which can occur, like there may be deletion of that particular gene, that there may be nonsense mutation of that particular gene, or inversion of that particular gene. So in case of inversion, there is complete absence of the factor 8 or factor 9. If the pathology is because of the inversion pathology, inversion means a part of the chromosome will be cut and get attached somewhere else in an opposite fashion, in inverted fashion. Suppose factor number 8, that particular one portion has been cut, and that will go and attach somewhere else in an inverted fashion. So that means if the inversion is the pathology, then there will be complete absence of the factor 8, because जो फैक्टर 8 जहाँ पे है क्रोमोसोम में वही एरिया अगर कट हो गया, कट होके दूसरी जगह में उल्टा होके ज्वाइन हो गया दूसरा क्रोमोसोम के साथ, ओके, so then there is no factor at all. Okay, so this may lead to this problem. Now there may be point mutation sometime which may lead to mild presentation with mild or almost a normal lifestyle. If the pathology is because of the point mutation, only one point is get affected. Chromosome is producing the genes are present in the genes, some portion, very minute portion is get affected, 1% or 2%. Then that particular gene go on producing the clotting factor, but deficiency is not much. If 100% deficiency, 100% affliction is there, then there is no more clotting factors are produced. But it is not like that, only 10% or 5% is affected. Then 90% continues to produce the clotting factor. So the problem will be either mild or moderate. Now these are the different pathologies related to the hemophilic pathology. So one is deletion. Deletion means, as I told, one part is get affected, deleted, and as shown in this figure, the deleted part is here. Similarly, this is inverse joining, very dangerous, which produce complete deficiency. So some portion is get deleted here, and that will be get joined in a different chromosome in an inverted fashion. AB and BA, it is written. And nonsense mutation, this is suppose a gene, and there will be totally mismatching. ADCM, I think you know the amino acid sequence, isn't it? Nitrogen bases. So that will be not mismatching. They are totally mismatching. That leads to nonsense mutation, And the last one is only one point is get affected, that is point mutation. So these are the different pathologies related to hemophilia. So will hemophilic patients always give the history of hemophilia? Now, as we know, this is an X-linked recessive disease, and the males are the sufferers and the females are the carriers. So we may get the family history of the male sufferers from the maternal side. Very, very important. From the mother's side, you will have some history related to this. X-linked recessive disease, males are the sufferers and females are the carriers. Okay, so now, so males are the carriers, females are all, males are the disease sufferers, females are carriers. That mother is always a carrier. But from the mother side, if any male, maternal uncle, if he is having the disease, because he is a male, isn't it? So he may get, so we may get some family history from the male sufferers from the maternal side. So it is true in most of the time. It is true in the most of the time. But sometimes hemophilia may be there without such positive history also. Sometimes you may get the history, but sometimes you may not get the history. 30% in cases it develops due to new mutation. Sometimes from this generation only the new mutation starts and the disease develops. This is possible in 30% of the cases. The rest of the 70% of the cases, most of the time you will get the history. Does the female also suffers with hemophilia? This is a question. Yes, at times females may also suffer with hemophilia. Rarest conditions, rare condition, but possible. How this happen? In the female, there is a process called as X inactivation during the blastocyst stage itself. See, suppose you imagine a normal female who contains normal X and normal X. Okay, but as we think, both the X are not active throughout the lifespan. Only one X is active, and that will go into what you say dominate over the other normal X. When female carrier, that means it will going to dominate the other. That means truly speaking, only one X is working. Normal, but still, just like two X cannot stay together, just like two females cannot stay together in a single house. Similarly, the two X cannot stay together inside the cell. Okay, therefore one of it will be dominant over the other, and one will be subsided, and another will be more dominant. So throughout the lifespan, only one X is active. Now, sometimes what happens if there is a carrier stage, and one X is mutant and another is normal, and if the mutant gene dominate over the normal X, okay, mutant gene will dominate over the normal X. The normal X will be subsided and mutant gene is dominating, it go on producing the genes and all. Okay, so now the problem can occur. As it is affected gene, it may produce deficiency of the clotting factor. So this is called as X inactivation process, and this is also called as Marie-Lyon hypothesis. Marie-Lyon hypothesis, she is the first who actually proposed this hypothesis. So now there is only one X present which is active and which is mutant. Okay, then what happens? The situation is somewhat similar to males. Okay, मेल में भी वही हो रहा है ना कि एक ही एक्स था और वो म्यूटेंट था और मेल में डिसीज़ आ गया. फीमेल में दो एक्स होने के बावजूद एक एक्स ने दूसरे एक्स के ऊपर डोमिनेट किया, और जो डोमिनेट किया वो अनफॉर्चुनेटली म्यूटेंट है, तो मेल वाला ही सिचुएशन आ गया उसमें भी. ठीक है, तो वहाँ पे भी एक ही एक्स है, वो भी म्यूटेंट है, एंड देन इट मे प्रोड्यूस द एक्चुअल डिसीज. Now this is exactly what is called as X inactivation process. Suppose she is the carrier female, XH and X the blastocyst stage, and normal X का inactivation हो गया. ये तो carrier था, normal X का inactivation हो गया और mutant X ही बच गया, and this is called as X inactivation. Okay. Now does the female develop hemophilia like males? Yes. At times the females may also suffer from hemophilia, so they are also called as symptomatic hemophilia in females. Father had hemophilia, mother has a carrier of hemophilia, suppose, then even the female may develop hemophilia. Father is already hemophilic and mother is carrier. So now in the female, suppose she has got both X's and one X is carrier and another one is a mutant gene coming from the father, so both are get affected, then the possibility is there that female can also develop hemophilia. When the homozygous mutation of the X chromosome is there, it can lead to hemophilia. So that is called as XHXH. H stands for hemophilia. So that is when father hemophilic and mother is hemophilic carrier. So these do occur when there is a consanguineous marriage. These are very common in the consanguineous marriage. Maternal uncle marrying sister's daughter, like that. So hemophilia with female with the hemophilia, this is a possibility. This is a carrier mother and this is father. Father is having the disease and mother is having carrier stage. So this is the possibility. Here the female is get affected. Female is get affected. To understand the hemophilia, we need to understand the hemostasis. Why this happens? The total pathology of this hemophilia that we will discuss in the next class. Thank you very much.